It is a short one, but a very impactful one. They have some fantastic speakers with us, actually. So this is held in conjunction with World Firefly Day 2023, which had the theme Show Off Your Fireflies, or Showing Off Your Fireflies, I should say. So for those of you who might not know, this is sort of also organized. This webinar is organized by Friends of Bukit Kiara, or more specifically, our Firefly Subcommittee, Magical Mysteries at Bukit Kiara, which is essentially a project that has been going on since 2021, studying the solitary fireflies that we have found in our beloved urban green lung at Bukit Kiara. So with us today, we're very excited to introduce Dr. Wan Jusoh, who is the Senior Lecturer of Biodiversity and Conservation at School of Science at Monash University Malaysia. Now, she's pretty amazing too. She's the co-chair of the Firefly Specialist Group of the Species Survival Commission of the International Union for Conservation of Nature. And she's in the steering committee of Fireflies International Network, where she serves as a representative for Australia and the Pacific Islands. Dr. Wan also has been featured in 2021 as one in the Asian Scientist magazine's Asia Changemakers, and emerged as one of the 3 winners of the 2022 TED Idea Search for Southeast Asia. Without any further ado, I'll pass the floor on to Dr. Wan, who will be sharing about glowing secrets, how fireflies get their names. The floor is yours, Dr. Wan. Don't forget to unmute yourself. We can see your slides. It's looking good, but just a gentle reminder to unmute yourself so that the rest of us can hear as well.
All right, sorry. Little bit of technical issue a bit.
No problem. I'm sure we've all had practice in Zoom, but it happens.
Yeah, although we have a little bit of practice already, right?
It happens to me as well, and I'm sure there are many people, for those who...
All right, sorry, because I kind of lost of Zoom just now, how to control Zoom. And then, yeah. Okay, hi everyone. I think I don't need to introduce myself. Thank you so much, Karen, for the kind introduction. And happy to see everyone here tonight. Thank you for sharing your time tonight to listen to me and my colleagues, my student as well, to talk about fireflies in celebration of World Firefly Day 2023. So what I'm going to talk about tonight is, all right. So we're going to talk about, but first of all, I just want to let everyone knows that this is about, so I'm going to talk a bit about what I do. And first of all is I also work on a species discovery, inferring extinction, and finally is actually to talk about, not to talk about, but mobilizing biodiversity data. I also actually teach these 3 courses: tropical terrestrial biology, global conservation and biodiversity, and human ecology and biodiversity. If you want to know more about my other work other than fireflies, so you please can go to the one for jusoh.com, and then you can see a bit more about the work I do. But tonight, what I will talk about is something about the glowing secrets. So in Malay proverb, it's called bagai kelip-kelip terbang malam. So it's actually if you look up on DBP side, you will see that it actually means open secrets. So if you talk about a glowing secret, it's already there, like it's already obvious, right? So then is actually talk about something that already common knowledge, right? So for a start, I will talk about, just give you a basic information, maybe a refresher about fascinating facts about fireflies. Okay, and then would like to go a bit more on the importance of scientific names. You might wonder why why can't we use common names? Why do we use scientific names or prefer to use scientific names? And lastly, I will introduce you to the interesting part about how scientists discover and name a new species. So in this case, fireflies. Okay, let's start with basic things. Fireflies are actually beetles. So I think even until today, after 17 years working on fireflies, there are still many people don't know that fireflies are beetles. They are not flies. Yes, of course, because of common name, right? Fireflies. Then you say they are not a fly at all. Yeah, so they are classified under the order Coleoptera and the family Lampyridae. So what is Lampyridae here? What is a firefly here? So you look at the head. So this is how we see them, is actually look at the big eyes, and then you see that there is a one that's called pronotum. So the good characteristic about fireflies is always to look at the, you know, the big eyes already hidden under this pronotum. And then they have elytra. Of course, this is a main characteristic of what you define as a beetle. It has a hardened wing that actually protect the other second wing, which is the soft wing underneath this elytra here. And then, of course, the other one, that what makes fireflies special from other beetles is their light organ in their abdomen. Okay, um, you must be wondering, right? How fireflies light up? Okay, so the start of this light show is this, what the special cell here is called photocytes. Okay, so if you magnify this part, so you will see it's actually something happening here, photocyte, in which can be found inside a firefly light organ. So what do you need is the recipe of adenosine triphosphate, the ATP here, and then the luciferin, and then the enzyme luciferase, plus some help from the oxygen. And then with that, you've got light. So you might also ask, right, why do they light up? Okay. All right, fireflies emit light to communicate. So first, it's as a warning signal to their predators that, you know, don't try to eat me. Okay, they are not pleasant to be eaten. So fireflies are not pleasant to be eaten. Okay, number two, is as a unique identity code, because different species has different flashing signals. So this is what they ask you actually. Hey, are we of the same species? And then thirdly, and the most important part is the for communication, sexual communication, where the male fireflies flash the light in specific pattern to attract the females. And then the whole mating process could take several hours at night. Okay. And then unfortunately, the mating and courtship process can be easily disrupted by light pollution. So this is something that my student Fathan will talk about it later. Okay. And then, so this is how the most, what we call it, the main important reason why fireflies give off the light. Okay. But here's the most important part, is also not all adult fireflies can fly. So if you can see here, there are many form of fireflies. They are all called fireflies, but as you can see here, male and female, there are some of them can have wings and can fly. And you can see the light organ by two lines of you see here, abdomen here, that has two light patterns. Sorry, not light pattern, light on the abdomen segments here. And then you can see also there are also female or firefly that cannot fly at all, and it looks more like a larva, right? But they are all glow in this group of species. Okay, so we think about why do they have different kind of fireflies. So it's not just one type of firefly, but we know from the morphology, it has a pronotum hidden, with the eyes are hidden under the pronotum. It has a hardened wing, and then it also have light organs. although not all of them actually are glowing. So these actually experts divide fireflies into several, what we call it, based on sexual communication or based on behavior. Here there are three of them: glowworms, flashing fireflies, daytime dark fireflies, or we call it diurnal firefly. So as you can see from this wonderful graphic by the Firefly Atlas, so you can see that the glowworms here, the adult male can fly, but the female cannot fly, and they looks like a larva here. And then they actually emit continuous green glow, not the flickering. But then the flashing glow, usually they give off the light that very discreet with the dots, like it's not the dots, but it's look like they blink many times, or there are flash interval, flash intensity. um and then both of them usually can fly. Both male and female can fly or have wings. But in this case of the daytime dark fireflies or diurnal firefly, the adult males, sometimes you can see their glowworm, their light organ, but they do not glow, and they actually active during daytime. But they are fireflies in this category, and they use pheromones to talk to each other. So yeah, we talk about the one thing I miss here is the life cycle. Okay, the short is 3 to 7 months, depending on species. So some species about 3 months, some species can be 7 months to even 1 year. If we are talking about Lamprigera, the star of the of a Bukit Kiara. And then the most important part or interesting part is larvae are carnivorous. So if you can see here, the fireflies spend most of their time actually during this stage, larva eating, and they eat, their favorite food is snails. Okay, in terms of number of species, firefly species so far, we have about more than 2,400 firefly species in the world. And this is based on the latest paper. And how about Malaysia? So I just put up this one pie chart. And as you can see here, the pie chart is it's not so accurate, but it's just to show you how much we know so far about our fireflies compared to North America. And then Singapore as our neighboring country. Singapore has about 15 species altogether, and Malaysia so far has about 50. And I would say that we could have more than 100 species compared to the other region is North America has about 150 species. So what happened here, that this is something that we want to talk about, is about species discovery. Why do we have, is it because we don't have enough species, but we are in a biodiversity hotspot? So we should have more species to be discovered. So you have learned some facts about fireflies. You also know that fireflies are known as Lamprididae. But you might ask, why do we need a scientific name when perfectly good common name is available, right? So let's take a look at these language, it's such a beautiful name. Look at that, like almost every language in the world has its own beautiful name for firefly. Like in our country, we have already kunang-kunang, kelip-kelip, and even api-api. And then you can see like other places has really unique name for to their country. But as you can see here, um, common names are often imprecise, misleading, right? Unlike scientific names, they can lead to confusion, right? About what animals is being referred to, and then what kind of relationship they are to other animals. So for example, here, a very famous example is about how Lampridae is called also fireflies, and lightning bugs are actually the same thing, right? So this is in the case of in the North America, in the US actually. And then also not universal in this case, Malay, if we speak Malay, we say klik-klik, kunang-kunang. It might be only known to people in Malay speaking region. How about the scientific names? So for scientific names, they are used to describe various species of organisms in a way that it is universal, so that scientists around the world can readily identify the same animal. If you talk about Lampridae, you know what I'm talking about. So you can describe by now. They are not flies, sorry. They are fireflies, they are not flies, they are beetles. They can glow, but not all adults can glow. They're not all adults can actually fly. So scientists from one nation can converse with scientists from another about a specific organism in this case, and so that it can avoid any issue arise from differing common names. So in this case, if you're talking about Colophobia brevis, because it has a standard what we call it binomial nomenclature, Colophobia brevis. So this is a quite common species in Malaysia. It's so scientific name is quite descriptive. In this case, when the name is called brevis in the Latin is what is for short, it's actually give you a clue about something about the last segment of the abdomen of Colophobia of this species actually has a short posterolateral projection. So that is what you have to read in the description. But this is it gives you a clue to what the species is about. But often, sometimes, because we are looking at the phylogenetic analysis, the relationship with the other animal. That's why some scientific names also sometimes change. For example, in the recent one is Nipponoluciola. This is used to be under the genus Luciola. So the famous Genji firefly, it used to be under the name Luciola cruciata, under the genus Luciola. So it was named Luciola cruciata until late last year. We changed it to Nipponoluciola after many data that we have collected. So naming is part of taxonomy. So you might have heard about taxonomy. Human, we are all taxonomies. We can call ourselves taxonomies. We classify things, right? We have chairs. We have call it these books. We have call it major, the chair, the desk, what we call as home, what we call as a car. There's a classification. So taxonomy is the science of identifying, classifying, and naming species. That is what naming species is all under nomenclature. So it's like life filing or library system. You are very organized, how you organize the life organism in this case. So that again, so that we can have a meaningful conversation when we talk about species, about biodiversity. So now we have learned that scientific names are important component in taxonomy. So you see that there are way of, you can still use common name, but there are times that is very easy, is very universal when you use scientific name. So let's say you have identified what you think is a new species. So it will require a name, right? So if I have a new species right now, how do I proceed? So this is something that I would like to share with everyone. So in this next slide, I'll be using examples of species and genus that my colleagues and I describe as new. So in my case, that involved me in 2015. So far, I only described 5 new species, and recently we proposed a new genus that I have just talked about for 2 aquatic fiber species from Japan. So that's one, the earliest one was in 2015. So I described these 3 species from Malaysia, one from Thailand, and the other one from Singapore. Okay, so step 1, well, you have to collect specimens. Well, when we talk about collecting specimen, it doesn't have, not necessarily has to be in expedition, in something like, we have to go field work, take a boat and go to some remote area. No, but it has to be also, it can also come from the museum, where there are a lot of things have been collected in the past, in the 19th century. And also it also can come from people who actually from donation, from personal collection. So in this example, that I went to with many agencies to actually collect the specimen. So after we collect the specimen, well, we had to get the specimens to a lab for processing and cataloging. Well, getting freshly collected specimen from the field to a setting that where we can carefully prepare, right? So sometimes when we have the specimens from museum, you should be prepared to loan it from the museum and then send it to your country. So send it where your laboratory is. So you have to be careful about the packaging. And also in this case, if you can see that when you are in the field, you have a sort of a good container to make sure that you also observe their flashing pattern and so on. Okay. And then step 3, I divided into 2. Step 3A, this is the longest time spent. So this is always people ask like, why it takes so long? But it's actually a matter of how the quality that you try to maintain here, the quality of species identification that you would like to do, or species information that you would like to gather. So examining specimen does not just involve morphological characters, but it's also need to have additional data. For example, in our case of Luciola singapura, we actually had to collect a molecular data from the species. And then we also designed the phylogenetic relationship so that we can also understand what is the common ancestor for this group, right? So, and also when people ask me, how do you do that? That look at the size of this firefly, that the finger. How small is the very tiny adult firefly. This one is Colophotia. And then you can see that I have to dissect this part and this part. So the abdomen here, and then imagine half of it is here, and then it's really, really tiny. So it really need a careful examination of and thorough in order to be able to describe what is the character, what are the parts of the important parts of the body, for example, to distinguish this specimen from others. Okay, and then step 3B is while you are working on that, you should be able to designate a holotype. So in this case, what is a holotype? So it's very important concept here. This is a species. When you want to declare it as a new, you have to say that this is the one that I refer to. So this is the name bearer. So if I call it Luciola singapura, the one I call it or I examine as Luciola singapura is the one now in Lee Kong Chian Natural History Museum, Singapore, and you can have a look at it, and then you can see that this is the preserved specimen that we use, and then you can look and refer to the description. So this is what we do. So for fireflies, so there are sometimes we preserve it in a form of ethanol, right? So but many important type specimen. This is important for you to know also that there are in the Paris Museum. This is because of the colonial practice last time when they brought back to Europe to actually deposit their specimen at the European Museum. So that's why most of our type specimen from Southeast Asia ended up in a European museum. So step number 4, this is the most interesting part. Everybody loves this, is name the species. So in the case of Luciola singapura, there are also, before I go into that details, usually what we call it etymology is the how do you name the species. So it depends on the diagnostic feature, like what makes that species that remind you like a Colophotia brevis that I showed you just now is because of the short projection, lateral projection at the abdomen of this species. But in the you can also name the species after a place. So like in this case of Luciola singapura, after some discussion, everybody will agree that it should be named after a country, the Kingdom of Singapura at the time, that we decide that it should be a Kingdom of Singapura. And then we can also name the species after someone, after some people. As usually in my case, I will name the species after someone who actually contribute a lot in the field of firefly conservation or taxonomy. Okay. Once you have named the species, you also have to make sure that you comply with widely accepted convention. In this case, is called ICZN. So the International Code of Zoological Nomenclature. You have to register the name to make sure that your name is valid. You can try when you have time. You go to ZooBank and then you can type in Luciola singapura, and it's already been registered under this one. So this means that you have already officially described it as a new species. And then finally, this is the last part of the naming us or describing a new species is you have to make it on official, like make sure that everything is written so that and then they publish, and then of course finally make sure also you deposit the new species the specimen in a collection. So in an established collection, in the case of Luciola singapura, is going to be is actually in Lee Kuan Yew Natural History Museum. So some of the type specimen are in a zoology museum of University Malaysia. Some also my whole like the types that I actually work on is in FRIM insect collection. All right, and this is a final one, but this is not does not apply to all, but this is what I actually practice. So it's also I also part of IUCN species so Firefly specialist group, where we actually advocate for the conservation assessment of the conservation status of species. And also I involve in Singapore Red Data Book, and they just released 2 weeks ago. So finalists of the checklist of beetles in Singapore with their national status. So Luciola singapura now being assessed as critically endangered. So this is something we can talk about it later in other story. But for this one, this is what we are working right now: assess, act, and plan. So we are making sure that we are just not naming it. But the most important part is once you have a name, you should, once you know about this person, once you know about this species, you should also think about what should we do about it. So I would like to highlight, so this is my last slide, I would like to highlight here is taxonomy is important. Taxonomy is important. I know some of you are out there will think about taxonomy is very difficult, taxonomy is very slow, but I would like to highlight here that taxonomy is important. We depend on taxonomy a lot to tell us the role species have in the ecosystem. Yes, naming a new species is time consuming, but any prospective new species must be identified, described, and confirmed before an appropriate taxonomy name is assigned. So this is also to help us to actually encounter how many firefly species can we find in Malaysia before they disappear. In the earlier, I already mentioned to you about we only have so far 50 that we described, 50 or 60 described species from Malaysia, but we should have more. But what happened is, can we still study new species before it's too late? So there are many potential of a new species is waiting for us. So this is just for you to, I will be happy if you will be, if you are going to be a taxonomy, if you are interested in taxonomy. But if you're not, it's okay. If you are enthusiast, you are naturalist, you can support conservation activities, volunteering as a scientist, like the Magical Mistress at Bukit Kiara program by FoBK, the one that Boon is going to talk about later. And if you're in academia, disseminate your research finding with stakeholders. And if you are in like what we do right now, and then what my student later Fathan will talk about his research, his recent research on fireflies. And if you are in policy making, you are an NGO working on policy, listen to experts and involve us, and also get involved in species assessment like IUCN Red List assessment. For that, thank you very much. Then I will I would like to thank FoBK for this opportunity to share my knowledge and findings about fireflies. And then I would like to acknowledge some slides actually reproduced from the video that we made for World Firefly Day 2020 by my friends Jolene and AJ Srimulat. With that, thank you very much.
Thank you so much, Dr. Wan. That was very, very insightful. I'd like to open the floor to the people. Please feel free to drop any questions that you might have inside the chat. And then we will be able to, Dr. Wan will be able to address them immediately. I think maybe on my end, if you don't mind, Dr. Wan, I have a bit of a question as well. Like, based on your understanding, I suppose, of what we know collectively across all the institutions or organizations or academia, as you mentioned, how much do we really know about fireflies in Malaysia?
We know quite a lot, but I think I would say that in terms of species diversity, number of species, like we are still counting. It's very, very few studies. We are quite stuck in discovering new species because of issues with the type specimen that I'm just talking about. They are in Europe, and then we need to go through the process. But in terms of the high priority species like mangrove fireflies, we are talking about Troptics, the one that people know about fireflies in Kuala Selangor. That one is Troptic stanner. Synchronous flashing fireflies is important in terms of ecotourism. People like it. So it has huge attention on it. So mangrove, and then on the other hand, is also critically endangered in this case. So we will give more attention to this habitat. So by having this kind of importance, we have a lot more research actually study on mangrove fireflies. But in terms of solitary fireflies, the other one that, or what we call it mostly dark time firefly or diurnal firefly or glowworm type that we have in Bukit Kiara, for example, like the one that is not, they're not in many numbers of and many individuals. They are not ecotourism potential. So that we have a lot of things that we don't know about them. I just give you an example. We still don't know the Lamprey-Gera, what species is Lamprey-Gera in Bukit Kiara. Yeah, so that is a good example of it. And also flashing pattern. I think that's also one thing that we don't have enough information. And at the same time, we also have to what's so-called race towards, protecting them because of the habitat threats and so on.
Okay, thank you very much for sharing. We do have one question from Clinton. He's asking, Dr. Wan, when was the latest new species discovered here in Malaysia or in Singapore?
Good question. Thankfully, I have that that information. Recently, actually, Singapore, the latest one is 2021, where I describe with my colleagues, Luciola singapura. So you can type Luciola singapura is quite well known in Singapore at the moment. 2021, that was actually the first time the luminous firefly was discovered, has been discovered from Singapore, which is like about 100 years. So this is that first time in 100 years. For Malaysia, recently we described a new species, several new species, not just from me, but also from Dr. Nada, from Prim, and Dr. Leslie Valentine. We had one monograph produced together that was in 2019. Yes, so it's quite recent. Yeah.
There you go. All right. Lots of work to be done on solitary, but we're very grateful, I suppose, for your dedication and commitment to the cause as well. Thank you very much, Dr. Wan.
Thank you.
Yes, okay. Maybe what I'll do next is introduce our second speaker. As Dr. Wan has mentioned, Fathan is actually her student. So Fathan is currently pursuing his research honors in tropical environmental biology at Monash University Malaysia. Fathan is also the president of the student-led initiative Green Representative Network, which is an organization which hopes to educate and promote environmental issues and provide opportunities for the Monash community to be involved. So his topic tonight is a pretty interesting one. Could artificial light at night be the cause of fading firefly populations in Kuala Lumpur? So great question, Fathan. We look forward to hearing what you have to share.
Yeah, thank you very much for the introduction, Karen. All right, let's hope I don't get the same technical difficulties.
It's a rite of passage.
Yeah, it might be. Everyone can see the slides, yeah?
Looks very good. Thank you.
All right, so I'll start. Hello, everyone. My name is Fathan, just as Karen mentioned. So today I just want to show you a bit about what I did for my research project, which was to find out if artificial light at night can be the cause of fading firefly populations. Just a very quick introduction. Unlike Dr. Wan, I'm not really as sort of decorated in some sense because I have just recently graduated. But so instead of explaining what I've done, perhaps I want to explain to you guys how I ended up studying fireflies. So I actually grew up in Indonesia, born in this beautiful sort of isolated place called Riau, Sumatra, completely forested. And one would think that that might be the reason why I started studying tropical biology, but actually that's not the case because I ended up just growing in Jakarta, a complete suburban place, no forest whatsoever from all the eyes can see. But then when I went to Malaysia to continue my undergrad studies, I just sort of feel very in tune and drawn to nature. And I've spent most of my time whenever I'm not studying actually out in the wild. So as mentioned, I'm doing my bachelor's. I've actually just finished my bachelor's and pursuing my postgraduate in Monash University Malaysia. And when I was actually figuring out what to research, I knew exactly that I wanted to do something that was environmental related, but not really know where to start. That was until I met Dr. Wan and we went out to Bukit Kiara. So for those who are not too familiar with what Bukit Kiara is, it's an old rubber plantation that was recently, well, not recently, but turned into this urban city park, right? Where they had these sort of paved tarmac areas, but also forest trails within. And during the day, it just seems like a normal, regular park. But when nighttime comes, that's when all these beautiful, magical lights just sort of come up. And these are exactly what you think, fireflies, right? And what's beautiful is the fact that there's, I think, 7 firefly species, and most recently, a potential 8. And the fact that it's so close to the city just sort of fascinates me, you know? And just by seeing all these sort of different lights, you know, so the variety. And here are 4 of, I would think, one of the more prominent ones, as Dr. Wan has just mentioned about its morphology. We have Lamprigera larvae, Pagaluciola, Colophothius species, and Parocelia. And you can see these differences in lighting. Dr. Wan mentioned just now about how they use these lighting not just to warn off predators, but also as a use of communication. So that it's a very important thing. And the reasons why all these fireflies have different lightings is so that they could, you know, identify themselves as different species. Just a little side note, at the very end, you see that this Regophthalmidae diplocodon, that is not a firefly, but is also considered a bioluminescent beetle. So this is why taxonomy is so important, because it's so you can identify, right? So this is not actually a firefly, but this really beautiful sort of star that has lights just glow in its entirety of the body, right? So upon seeing this, 2 questions popped up in my mind, right? Firstly, with fireflies, you know, relying this bioluminescence at night so importantly, does that mean that artificial light at night from human sources could affect these fireflies? And if they do, being so close in proximity to light pollution from the city, does that mean, is there a potential that we will no longer be able to see fireflies if these sort of lights continue to encroach? So these 2 questions were exactly the questions that I answered during my final year project at Monash University for my undergraduate for this unit called SCI 3990, where I spent about around 3 months along with my friends studying fireflies to answer this very question. So before we continue on, I feel like it's important for me to explain to you guys what exactly is artificial light at night. Well, it's essentially any source of artificial light that occurs at night that can affect the natural sky. And it can come in 2 different ways: direct illuminance or the scattering of light. So when we're talking about the direct illuminance, we're talking about in short distances. So lights that come out of your windows at night, or street lamps, or even billboards. Well, the scattering of light, this is where it gets a bit more interesting, is that it comes from the diffusion of light from a very concentrated source, for example, city lights, that would radiate out into the sky and then sort of gets reflected by the atmosphere and the clouds. So this creates this sort of sky haze. And one of the reasons why, if you live dead right in the city, you won't be able to see stars. And this is something that we call sky glow. And this becomes a very big issue, especially when we're talking about fireflies in urban areas, because with the increase of urbanization, the increase of cities, that means the encroachment of these lights onto the fireflies, right? So talking about the impact, how does these lights impact essentially species that rely on, you know, light? In 3 ways: spectrally, temporally, and spatially. Spectral, I'm talking about essentially the wavelength of light that artificial light will impact the natural sky. So if we're talking about, for example, colors, if the natural sky is, for example, very dark black, if we introduce, for example, blue light, it could actually change the hue and make it look different, changing the spectral composition of the light. Next, we're talking about temporal and spatial, where it's temporal meaning the time of day where natural light exists. And lastly, spatial is, as I've mentioned, the amount of space of natural light that retains. And for biological species that rely on light, especially like fireflies, for its source, for a resource and source of information, this is unsurprising that it could impact them, right? So how does ALAN or artificial light specifically affect fireflies? Well, two different ways. Its mating success, as Dr. Wong had mentioned, and also its nighttime activity. So how it does this is that when there is a source of artificial light, it can mask the bioluminescence that is coming out of these fireflies. So when a potential mate tries to find another firefly, it could actually make it look almost invisible or make it less attractive, hence reducing its possibilities of mating. And secondly, is its nighttime activity, which could be said to be the most important, because how it does this is a lot of fireflies are nocturnal or crepuscular, meaning that they are active during darker times of the day or days after sunset. And what happens is a lot of these fireflies actually rely on the sort of natural sky to see the changes when it comes from day to night. So with the source of artificial light, these fireflies won't be able to see these changes, hence reducing their active time during the night. And also because of the spatial space that these artificial lights are encroaching towards, it also reduces the amount of space where these fireflies could be active, right? So for my project, we did a very simple survey. This is something that we call a transect sampling, where essentially we just walk from one end to another, right? And throughout the entire path, we sample anything that is within 5 meters of us. And how we sample is using this method called capture and release, where we try to capture whatever it was and then release it after we've marked it and took its GPS coordinates. And the area that we chose is this tarmac area within Bukit Kiara, which they had street lamps lined throughout it, right? So we can actually have the street lights either turned off and on. And what we want to see is also whether distance is an important thing, whether we would see more fireflies the further away it is from the lights. So what we did was also measure the distance between each street lights and the occurrence of each fireflies. And during the survey, it's actually a very simple thing that we did was, I feel like anyone can do this because all you takes is sweep nets to capture the fireflies, specimen vials to keep them in place and identify them, or featherweight forceps to pick up those larvae that are usually around. And one thing to note is that we used red light almost the entire time of the day, because some studies have shown that fireflies are actually almost blind to long wavelengths. So that is light that is closer to red. Hence why we use red light during our surveys. So what we found was exactly what we thought. So here is the results. As you see here is a heat map that I've made where each blue color, the colors on the map are essentially the frequency of fireflies that was observed. On the left is when the streetlights were off, and on the right is when the streetlights are on. And each dot that you see are these streetlights. And from here, you can already clearly see that when the lights are off, you can kind of see that there are already fireflies distributed around the area, while when the lights are on, immediately a drastic change, a drastic drop in fireflies were found. And one thing to note is where the fireflies are distributed. If you look at the one on the left, you can even see that some of these fireflies actually overlap with where the streetlights are. While if you look at the one on the right here, those are a bit far off away, a bit sort of away from the lights, right? And we look at the statistics. This was exactly what we found. There was statistical difference between the abundance of fireflies when the lights are on and off. But what's interesting was we thought that the further we are from the lights, the more that we'd find fireflies. But this was not the case. There was actually no difference between the two. And instead of this sort of like increasing linear line when the lights are on, we actually see this up and down, right? Which shows that perhaps distance isn't exactly what's affecting these fireflies. But if you look very closely here at the very first view, you can see that there were no observations of fireflies when the lights are off. But you can see them when the lights, so this shows, right? If you think about what the main reason of streetlights are, it's to illuminate the immediate area, which shows, right, that fireflies still keep away from lights that are of direct source, meaning that perhaps when it comes to these sort of occurrence of fireflies, it's less relation to how far it is from the light and more to where exactly it is. So it's more about the microhabitats and where you find it rather than the distance. And you can actually see it right here. This is a heat map of the firefly observations when the street lights are off. So here's the thing. If the lights are off, we assume that then we would be able to see fireflies across the entire tarmac. But that's not the case. There are some areas that I like to call areas of isolation within within the tarmac where we found no fireflies whatsoever in all our surveys, right? So for example, here, if you see here, this first stretch of line, no fireflies at all. And if you look at the surrounding areas, you could actually explain why you might not be able to find it. For one, you can see this super bright, and there's very few sort of cover to sort of block out all the lights coming from the guardhouse. And if you look at this second stretch, right, again, almost no canopy to cover sort of the sky glow coming from the night. And also, there's a high level of human disturbances where you can kind of see here a little human built, and then a lot of built up areas, which may explain that maybe these human disturbances could be the reason why we don't see fireflies. And one very interesting thing here again is these two areas, right? This one, open clearing that you can see immediately into the city sky. And this one is this long stretch of road where there's this constructed area onto the onto a mountain that's paved in white, right? This is to prevent landslides. But if you ever happen to come here at night, this specific area is very bright because this light coming from the city is reflected onto this white surface, making that entire area bright. But if you compare this to this stretch of road here where we see a high number of fireflies, here's a photo that I took, completely dark, and you can see this high levels of sort of coverage to keep the area pretty much away from light. So in conclusion, what we found was exactly what we expected. When there is a presence of light, there is less fireflies to be found. However, when it comes to distribution, it doesn't seem to relate to distance, but more relation to the environment, where they may be found much higher in darker areas or areas that are blocked away from artificial light. And what this might imply is that further encroachment of artificial light at night and development in Bukit Kiara can continue to decrease the firefly population. But there's actually some hope, because like we see how when the lights are off and on, when it was off and when it was on, we see that there's very few fireflies, but the next day when we turn it off, fireflies will come back. So this might actually show that there is hope, that if we decrease the levels of artificial light within Bukit Kiara, this may improve firefly populations in the future. So a very quick is that this is a very quick sort of intermediate study that I did that I only did in 3 months' time. A firefly larvae life cycle can last almost 2 years. So there's a lot of limitations and future work to be done. However, it's not something that one person can do, I feel. and it takes an effort coming from a lot of people. So this is where I want to sort of plug in also the Citizens Science Program, the Magical Mysteries at Bukit Kiara by FoBK. These sort of programs actually help us understand more about fireflies, and by having citizens help sort of get these data, we can actually exponentially make these process of learning more about fireflies much faster, which I think Boon, our next speaker, will be talking a bit more today. And that's everything. Thank you.
Thank you very much, Fathan. Hold on a sec, let me put on my video. That was fantastic. And I guess it kind of confirms a lot of the suspicions that we have. So thank you very much for sharing on that. We do have one question from Dr. Rai. Would the leaf litter on the floor adjacent to the tarmac be a factor that influences the abundance of the fireflies as per your distribution? And the second part would be, what would be the other confounding variables?
Okay, yeah, 100%. Because there were instances, right, when I was surveying during the night and we had all the street lights on, right? There was this street lamp, right? Super bright. And then there I found, just within a meter from the light, is this Lamprigera larvae just hiding under a leaf litter, sort of like keeping the area dark. So 100%, I think leaf litter adjacent to the tarmac is a huge influence to the abundance of fireflies. When it comes to a lot of confounding variables, there's a lot of them, right? One that we had a lot of issue, for example, is when it comes to weather. Fireflies love moisture, but at the same time, if it's if it's still raining, it's still a very bad time for them to come out, especially when it comes to fireflies, right? It's a bit hard for them to fly around when there's rain. But after the rain, that's like the beautiful time for fireflies to come out. So during the time when I was sampling, it was during the rainy seasons. So it was raining almost every day during the time that I was supposed to sample. So that became a huge issue. Secondly, for example, is moon phases. When we're talking about looking at the effects of Allen, right? Because different moon phases will reflect the levels of sky glow from the cities differently. Secondly, also cloud cover. But a lot of these confounding variables could definitely be helped with maybe further use of more equipment, like SQM sky quality meter would really help. There's a bunch of other confounding variables that we definitely could look into is, for example, microhabitats. What sort of microhabitats that fireflies prefer more, right? So yeah, this is where we're talking about, like, there's so much that we still don't know about, so much that we still have to learn, and there's so much that one person can do that I think having citizens actually be part of this could really help.
Thank you so much for that. We also have another question coming in from Tasha, which is, do you feel like the light from the street lamps affected how easily you were able to spot the fireflies compared to the trial without the street lamps on? Sorry, that's from Rhea, not Treya. Tasha.
A very good question. Yes, there is 100% possibility as part of a one of the the underlying factors. It's very possible that when the lights, but actually, right, when the lights are on, it's much harder to see the fireflies, because again, that's where we were talking about the masking, right? Because the presence of other lights actually make it harder to spot the firefly lights. It's actually easier when the lights are off. So there's definitely ways that we can make this study better, perhaps using our own source of light. And instead of doing a for us, we did this transect line sampling, right? Maybe we can do a point sampling where instead of moving, we just stay in one direction to make sure that we spot everything that we see, and then wait within maybe for an hour or so within that one spot. But it's very time consuming, which is one of the reasons why we weren't able to do that. But yeah, very good question.
And just maybe a personal question for you as well. Have you decided what you'll be doing for your postgraduate research?
Yeah, honestly, I was so fascinated doing this 3-month project that I feel like there's so much potential that could be done, that I would love to explore this more, expand this Allen study, this artificial light study, and look into other areas of Bukit Kiara instead of just this one spot. I think there's so much to be done, and so much possibilities. That just
Thank you.
yeah.
And just before I introduce Boon, we have one question from Dr. Cribben as well about if he was given to understand that changing the wavelength of the light may help the firefly populations. So are we any further in our knowledge to recommend changes to the lighting to be used in Bukit Kiara?
Okay, yeah. Yes, I've seen studies that show that using red wavelengths does help, because they seem to be almost blind to red wavelengths. But one thing I've noticed from that study is it might very much be species specific, because there are other studies that show certain species of fireflies actually don't show the same characteristics. So when it comes to knowledge, we can recommend changes, but I feel like for us to be 100% sure, I think doing an in situ study to be 100% sure about it could really help. One thing that I've been thinking about doing is perhaps installing our own sort of sources of lights instead of relying on the street lamps. And that way we can use different wavelengths and see whether fireflies in Bukit Kiara are affected by red wavelengths or not. But yeah, 100%. It is potential, 100%.
Good to know. I mean, I believe your work will hopefully bring changes down the line when it comes to like urban planning and urban lighting.
Fingers crossed, yeah.
Fingers crossed. All right. Okay, we've got our next speaker. Last but not least, we have Han Boon Hua, who is a career geologist as well as a hobbyist naturalist. So Boon, if you've joined any of our Bukit Kiara activities, specifically the Firefly activities, this is the man who has been running or co-chairing the Firefly subcommittee of FoBK since 2022. Pretty much coordinating everything Firefly related in our project, Magical Mysteries at Bukit Kiara. Now, he'll be sharing many fascinating things, which will expand on what Fathan and even I think Dr. Wan shared. Without any further ado, Boon, please take it away.
Hi, I've got to do this customary question. Can you hear me?
Very well.
Okay, cool. All right. Okay, last but not least, thanks everyone for joining. Really appreciate your time on a weekday, nonetheless. And thank you, Fathan, for lining me up nicely into introduction of our citizen science program. You've done a lot of heavy lifting descriptions. Appreciate it very much. So allow me to continue, where he left off. Okay, so what I'll do here is I'm gonna give a very brief history of what FoBK is about, a little bit more about how the Fireflies encounter pan out over the past 5 years or so. Types of fireflies, Fathan's already covered that. Brilliant. I might touch on that a little bit more. I'm going to share a bit more about the kind of surveys, citizen science-based surveys that we are doing. Share some very prelim results, some statistics, and also share with you, what are the small impact that we think that we are able to exert, and challenges, and then what do we want to do next? Yeah, moving ahead. Okay. All right. So we are friends of Bukit Kiara. A lot of you may have already known what we are doing, but I do see that there are quite a lot of new names, new people in here. So we are a non-profit based in Kuala Lumpur. Our objective is very specific. It is to pursue protection gazettement of Bukit Kiara, which is a green space in Kuala Lumpur. I'll have a bit more details on where it actually sits a bit later on. But essentially, what we are trying to protect is this area with about 400 odd acres of a rehabilitated secondary forest. lots of good stuffs in there that we are continually, learning about, and we want to build appreciation, of the community towards this little island of a gem that we have in the city. And the Firefly subcommittee is one of the many volunteer-driven interest groups within Friends of Bukit Kiara. So anything Firefly subcommittee is there to carry things out. Yeah. So Firefly was first reported. Yeah. Now people first paid attention to this thing that shines and flies in Bukit Kiara back in 2017. So Bukit Kiara is already quite a popular place among city dwellers for hiking, mountain biking, trail running, and whatnot. And then in 2017, Fireflies. This predates me joining the group, and based on what I've learned, people were actually quite clueless. They were very excited, but they did not know what they did not know. So the organization, the people in there started reaching out to, naturally, Malaysian Nature Society, starting to build an understanding of what exactly is that, what creature is it, right? And then this culminated a year later in the first public mass walk, and it received overwhelming response, like literally overwhelming. So you got a walk and 100 people turn up. Yeah. So this overwhelming response, spawned this idea of perhaps we should do something that's more sustainable, that will last, that they will have longevity. So this idea of a citizen science program was then hatched. Started training. What we call Firefly ambassadors is just a glamorous name of our volunteers, our regular volunteers. We equip them with some knowledge, and then let them build muscle memory. And then we also started engaging with stakeholders, like in this particular example, our member of parliament of this area, which in a nutshell helped a lot in our conservation work here as well, right? And then things really took off in 2020, right before COVID. So FoBK engaged specialists within KL who are more well-versed into the world of insects and fireflies. So we have Forest Research Institute and also UBI. And we started devising a protocol, a basis to identify or to understand what exactly are these fireflies that we have in Bukit Kiara. And also the good work of the predecessors back then, they secured seed funding from UNDP, SGPGF to actually kickstart this program. And with that, we officially began. So the Magical Mysteries program officially began. And so we scale up training. We get a much bigger Firefly ambassador pool. And then, of course, COVID hit, but that didn't stop us. And then, in fact, COVID helped, people to realize that, well, they actually they appreciate, nature a lot more than they used to, because they have built this reliance on nature when everything else, disappears, right? So, so the work continued, and the initial seed funding lasted a year. Right, so that was supposed to be it. We got a bit of a baseline data, but the group, the core group that did the work, decided that we should continue. Yeah, so we've built up a lot of muscle memory, a bit of knowledge. We have support, and then we should keep going, and that's what we did. So, so since then, we have continually, conducted our surveys. And then recently, this year, we've also started working with Monash University, where the students came in not just to join our citizen science program and surveys, but also to conduct, studies like what Fathan has just presented earlier on. So we would like to go from strength to strength going ahead. Okay, types of fireflies won't bore you too much. This is not the point of this talk, but essentially we have 7 species, and in fact, we recently we found 8 species, right? Diurnal species. I'll come to that later. So our flagship is Lamprigera, quite a large firefly in terms both for both the larva and then adult. And I have a side story to tell when it comes to the Lampreygira, specifically the male. So with the first specimen that attracted attention really was the larvae. Yeah, so it caught people's attention very, very quickly. And once we started going out and surveying, we've also encountered the female adult, but we've never really seen the male. And so we started the group, or the scientists supporting us also started framing hypotheses of what may be. Until one fine day, on a group in Facebook, someone posted this picture, a bit of a post, and it was like a bingo moment. And then almost spontaneously, the group launched a search party. Yeah, so we look at what time. We contacted this gentleman, find out what time was it found. Did he encounter this particular specimen? We went out, and after a few attempts, we managed to track this down. We managed to encounter the male, and incidentally, it is crepuscular, which is why we never, found it in our normal surveys, because our surveys are done like after 8 p.m. And this guy literally, comes up within a half-hour window at the sunset. So very fascinating. And yeah, so allow me to go back to the other species. So it's Lampreygira is quite big. Colophortia, which is on the other end of the spectrum, quite a small specimen. How small? You got to come out, and we will show you how small it is really. And we also have Pyrocelia, Luciola dumona, the only one with the species that's identified. So alluding to what Dr. Wan was mentioning, so first identified, the sample that's identified first or actually came from Boon in Trungan. Yeah, Diphanus. We also have a Drilester and Stenocladis, which are diurnal in adult form. We do not know much about them. Yeah, and last but not least, the eighth one I was talking about, Lucidina. So another side story. So we had our Earth Day celebration about a month ago. And yeah, we were out there in Bukit Kiara, in the hills, setting up stalls, pop-up shops, things like that. And, our volunteers started, seeing this little guy flying towards our people. And then we got our experts then and there, and then we started, joining the dots. 1 + 1 is 2, and then there you go. We actually have another species identified during the day. Very auspicious indeed. All right. And then on a side note, there's also this Ragophthalmidae, or more commonly we call them the starworm. Very, very fascinating, which we also encounter them in Bukit Kiara. So in a nutshell, quite a lot to see. Yeah. So as promised, I was going to tell you a bit more where we actually do the survey. So we actually run different type of surveys. They are graded according to difficulty. Yeah, so a little bit about that later on after this. So essentially, we have these dotted tracks, which are tarmac paths, very comfortable, very easy to traverse. And we also have the more challenging nature trails, which we go out to conduct some of our more difficult surveys. And where we are, Bukit Kiara, it's right here. So you can look at this blue line down here. This is 10 kilometers. We are literally less than 10 kilometers from the Twin Towers in downtown Kuala Lumpur. So, and we are an island, yeah, in this sea of a concrete jungle. And yeah, that essentially sets us up, yeah. So, as I mentioned, 3 types of survey. Yellow survey, it's done in the day to collect environmental parameters. You probably won't see any fireflies, but now you know a bit more about diurnal firefly. You may encounter them after all. And we take measurements, environmental measurements to supplement, the count that we actually do for red survey, which I'll come to in a short while. The orange survey is a capture, mark, release, recapture exercise for only Lampreygira on the tarmac path. And we also measure the width of the larvae for every single specimen that we encounter, right? So in terms of difficulty, we are 1 notch up now. And finally, participants will graduate into the red survey, where we go out into the nature trails. It is essentially a count exercise. So the skill set that we require is identification. So participants, having gone through this, we have also prepared lots of materials, we can then help volunteers to try to identify the species that they see in the trails. And that's done on 4 nature trails. So all of these are done at least once a month. Yeah, so pretty regular. All right, so yellow survey, so a bit of an idea of what we actually do, setting up a quadrant, soil meters to take measurements like pH, temperature, etc. A good old ruler to measure leaf litter thickness, leaf litter, which is very important component for the ecosystem for firefly. Yeah, and we also take a lot of pictures along all the different sides of the quadrant. So this is specifically to capture vegetation density. And we also take pictures of the canopy cover. So the idea is to eventually digitize all these pictures into data points, which we can then use for analysis, cross-plotting, for all sorts of numerical work. Okay. Orange survey, which is a night survey for Lampreygira. So this is the bit where we actually measure the width of the sample, and this is the marking that we are doing. Okay, and then red survey. So I'm just putting out here an example of the month, or the data collected from the month of September, right, in 2022, and we've actually plotted all this data in pink, the count data, into a map form. So and we will essentially digest this, process this data into a format where we can extract count information of the different species, whether they're adult or larvae, so on and so forth. So a lot of good work that our volunteers need to do. Okay. So why all this? What do all this data mean, right? So, by themselves, the surveys actually provide an avenue for people to get out, to get in touch with fireflies. So, in our outreach work, we considered that one of the objectives that we want to achieve. But to those who are more technically inclined, numerically inclined, data inclined, we actually try to make use of the data that we collect. So in this particular case, this is data, distribution data of the 7 different firefly sampled between March and 21 to February 2022 in a spatial sense. So we can start to form an idea of where certain species congregate at, or maybe it's their natural habitat, and find correlations for reasons of them doing so. And then we can also look at the data in terms of count by time. So what you are seeing here is a fluctuation of the firefly population, in this case Lampreygira larva, across 2021 to September 2022. So in this particular case, it forms a baseline. So whether you think that it's coming down, or this is natural fluctuations, that is a bit where statistics come into the picture. Okay, so some highlights, some stats. So far, until this month, we've got 279 citizen scientists that's gone through our system. These are the number of active ones. We have quite a few of junior citizen scientists as well, right? And out of this, we have 26 that are really our core group. Kudos to you guys. These are the guys that do not just help to lead our firefly work, but they are also in the subcommittee, running things, brainstorming, moving things, think about what to do next. And we really, really appreciate all these volunteers that's been with us on the journey for more than 2 years, actually, a lot of them. Oldest one, 74 years old, the youngest one, 5-year-old. Okay, and number of surveys conducted. Since November 2021, we got 24 yellow surveys. the 35 orange surveys. So on average, if you think like one survey a month, so that means that that's about 2 years worth of surveys. And for red, we actually have 61 surveys covering 4 different trails. So yeah, we hope to sustain this, go from strength to strength. And along the journey, we've actually made incremental improvements thanks to the volunteers. So initially, when we first started, this is actually a Google spreadsheet, Google Sheet, which we use in the field on our volunteers' phone to actually input all the data. And then we got some pretty smart programmers in our midst, and then we started using web-based tools to then automate this process, which really helps to increase the speed of the survey and also to help us collect the data at the back end. In terms of the measurement, so I was telling you about this caliper. It is a very painful exercise. You got to do this. The larva is not particularly huge, and for statistical consistency, we have to take 3 different measurements for every single specimen which moves. And then someone started moving this idea. We should probably take picture on a grid and then we digitize it. So this is what we've done, and we have baby steps in trying to digitize this information. So one of the mini projects that the Monash students are doing, but it's exactly this, to work on digitizing this data set into something numerical which we can use. Yeah. So what impact have our surveys brought? We've discovered 7, recently the 8 species of firefly in Bukit Kiara. So for 400 acres in the middle of the city, we think that this is pretty amazing. We've got a much deeper understanding, and particularly like for instance, the Lumbrigera male, which I mentioned earlier on, the kind of behavior which you only really get when you dive deep into this particular species in a recurring basis. We think that we've established some form of baseline count and also environment parameters. So we want to continue, keep going, and hopefully, then in the future, when cyclicity kicks in, then we can start to track changes over the years and decades. May I dare to dream to come? Yeah. And also one particular small win that we think that small impact that we've exerted is actually addressing a leaf litter management within Bukit Kiara. So the Bukit Kiara it's an arboretum, so it's like a park, a natural park. But a lot of the leaf litter practices were exactly what you would do in a well-manicured garden. Yeah, so a lot of blowing of leaf litter, especially adjacent to tarmac paths. So we pulled out the data from back then. This is from 2022, and then we started noticing this drop. So we weren't sure, statistically, we weren't sure whether it's significant, but we are seeing this downward trend. And this actually formed the basis in a shift for the park administrators to actually, go easy on their leaf litter management practice. And this is unofficial yet, based on a prelim analysis of the data, subsequent data from that. It appears that there is an uptick, but we shall, keep you guys informed when we have into more data, more analysis, and yeah, in future we will share, everything that we learn. All right. So, what are the challenges and how do we move ahead? So, the first thing is to continue to spread the word. So, even when we talk to people as close by as in TDI or even Bandar Utama, just adjacent, we are literally like 2 kilometers from Bukit Kiara. There are still a lot of people who are ignorant to the fact that there are fireflies in Bukit Kiara. So our challenge is to continually, spread this word, this knowledge. But to do that, we also need to grow our pool. We need to recruit passionate volunteers, people who cares and who's willing to put in, some miles in doing what we are doing. There is time involved, definitely. So for instance, a lot of the data processing that we do requires time away from our day job. And so in terms of data processing, we are continually lagging behind the data that we acquire. Technical know-how is it's also important, but I think we are blessed that within the Klang Valley, that there's always someone knowledgeable that's with us on our journey, and we really appreciate that. So and we want to keep going with this. Yeah. And then there are certain species, like for instance, Pyracea, the female, we've never encountered a female species. So what's going on? So this is like the male Lamprigera, problem all over again. So this is definitely something that we can tackle going ahead. The fireflies that we can observe, most of them are come out at night, nocturnal. But how about the diurnal firefly? So I was giving the example of the Lucidina earlier on. So do we know much about them? That we probably do not know. So there's definitely a lot of room to study, species like that. And perhaps, alluding to what Dr. Wan was mentioning about, probably the fireflies in Bukit Kiara, there's potential to put them on the IUCN red list, So there's a lot of work that needs to be done, to assess this. So this is one of the scope as well. So we really want to continue to sustain and strengthen the citizen science program, continue to acquire, data, process, analyze, like some of the longest running citizen science program, like in Carolina, in America for bird watching. Yeah, it's been going on for 136 odd years. So why not us? Yeah, we are only 2 years, barely 3, into the program. So we got a long road ahead, right? So acknowledgement, a lot of people to acknowledge. On the way with a lot of angels that helped us. We couldn't stress enough on how important the people that we meet along the way are. The community, the subcommittee, you guys really rock, and please bear with us, and we will continue. And we hope, to those out there, if you have a little bit of curiosity, do join us. We welcome everybody. All right, thank you.
Thank you so much, Boon. I think that was a very, very good intro. And I think it'll be fascinating as well to see with what Fathan has plotted out on his distribution map, if we see similar things also in terms of the Lamprigeras we spot on the tarmac, which are lights. It will be quite exciting to see that. We do have one question coming in from Dr. Rai. Besides the firefly species, is there also works on looking at their diets? So this could be potentially significant to further enhance the conservation efforts of Bukit Kiara, for their diets could also be of the same status as of the firefly.
Yeah, very good point, Dr. Rai. We do actually also, whenever manpower permits, we try to also conduct a snail surveys concurrently with our red firefly surveys. So snails being the major food for firefly. So yes, we do collect that data. And now the devil is in actually utilizing the data and make a cross comparison. So we do endeavor to collect this data, but when push comes to shove, when we don't have enough personnel for a particular week or night, well, the snail survey is the one to get dropped, unfortunately. But yeah, we do try to collect this information to try and understand it.
So this is again invitation to all who are listening in either via Facebook or if you're in the Zoom call. And of course, if you're based in Malaysia, please come and join us for the Citizen Science Project. It is completely voluntary and it's quite fun. You meet a good bunch of people and you would be able to advance the cause for science. Well, I just want to see if there's any last questions from anyone. Feel free to drop it in the chat, whether you're on Facebook or in Zoom. We have actually, okay, thank you so much, Adeline. We have a question here. So there are 3, each speaker actually has a question from Adeline. So perhaps maybe Boon, while I have you here, would you like to tackle Adeline's question first, which is just wondering, has Bukit Kiara been fully explored, or how much of the area is there to be explored?
Bukit Kiara as a hiking trail locality, I think it's pretty well explored. There are more than 100 trails in there, so there's a lot of knowledge in there. But when it comes to firefly survey, we're barely scratching scratching the surface. So there is definitely a lot of room to scale up, like even Fathan also mentioned that in his talk, do studies on other trails, other parts of the park. But the issue is always manpower, so that is our biggest bottleneck. So as it is, just to run our normal surveys a month, right? I think we would really need about I would say 6 to 7 leaders, people who are the Firefly ambassadors, who are really experienced, and then we'll bring in anywhere between 5 to 15 volunteers, or maybe even 20 volunteers a month, and that's only for our 3 different surveys across an aggregate of maybe 6 trails. So you can imagine if you want to scale this up, right, without creating fatigue for the same set of volunteers, we really need manpower.
True. Always, I'm sure, perhaps Anne from Justice, JWM also would agree. Volunteers are very crucial, backbone of a lot of NGOs. Father, one question for you. What's the ultimate goal of your Allen study? Do you know if Allen has an impact on the larvae of fireflies, or is your study mainly focused on flying adults?
Thank you for the question, Prof. Adeline. I'll first tackle the second question. From when I did my survey, it was actually both on fireflies, larvae, and adults. And I found that both larvae and adults are affected by Allen, but one not as much as the other, because the reasons why they employ light is very different. For example, for larvae, it's mostly to ward off predators, while when they become adults, it sort of evolves more into more complicated ways of communication and courtship. So in some ways, we can say that a light is more important when it comes for adults, but still equally important for larvae to protect themselves. When it comes to the ultimate goal of my Allen study, mostly for my own benefit of learning more, because I am generally curious about delving deeper into this, but it would be great if this study could actually be used for real life consequences, because I think a lot of us who are doing environmental sciences have this sort of want to make the world a better place, and having positive changes using scientific backings would really, I think, be great. But yeah, thanks for the question.
Okay, last but not least, we also have a question for Dr. Wan. Just out of curiosity, are there more female fireflies than male fireflies?
Okay, thanks, Adeline, for the question. Actually, I cannot say for sure, but what visible to our eyes, we see more males. For certain species that we can see visibly, like Pyrocelia, not Lamprigera, even Lamprigera. Actually, Lamprigera we see more of larvae, right? But then for the Luciolini, this subfamily, if you see the species, you can see more female actually flying around. So it also depends on the way the sexual communication, the way they talk to each other. So that's why I learned. I just now I talk about 3 types of fireflies: glowworm, duck-tongue fireflies, and flashing fireflies. But for flashing fireflies, the mostly I will say like quite equal number, because they need to communicate, like give response to each other. That's why it's visible to our eyes as human, right? But for Lamprigera, which is like the female actually glowworm and like to hide, and probably maybe hidden under the leaf litters. We won't be able to see it, but if it's quite rare for us also to see the male of Lamprigera as well, because of the time that they appear, which is they have this sort of behavior, like we call it crepuscular. So it's very hard to answer this question. But if you talk about congregating fireflies like the one in Kuala Selangor, you see like all of them are together, they're actually quite a bit, like quite similar number. You will say that the ratio is a little bit about the same. So because so the answer is going to be dependent on the species, or even the behavior, or the way they talk to each other, the communication. Yeah.
Just want to say one thing. Boon mentioned that that FoBK hasn't found any female Pyrocelia, right? male paracilia. Very interesting thing was during one of our surveys, we actually found a mating paracilia
Oh,
by the office trail. So very fascinating.
you should, you should be maybe like a take note of the coordinates. So it's very hard about this
Mm.
group like Paracilia, Diphaene. So this group under the subfamily Lampyrinae, actually very hard to spot the female because the female usually flightless. So maybe we cannot see them, but they are just like, hey, I see you guys, but they are really good at covering their tracks. So hopefully that's the I hope maybe next discovery, next work to do with Monash students and FoBK is to explore more about the other parts, the other mysteries of fireflies in Bukit Kiara.
Very cool. Thank you. Boon, did you want to say something?
Yes, while we still have the forum here, and because we got quite an audience, because I think towards the end of my talk, I dwell quite a bit about, I think I probably shed some light on what we think we want to do, how we want to move ahead. So I did focus a lot on the volunteers aspect because this is a citizen science program. But there's something that we are currently exploring as well is to actually obtain funding in the sense that we would our core task is still on the survey aspect, acquiring data, but we are starting to explore whether it's possible to take out chunks of the data processing and analysis work, but this requires either hiring manpower on a part-time basis or outsourcing it as chunks of projects, but all this means that we need resources, so we need funding. So to the people out there, if you know of any organization or anyone, any philanthropist that's really into, keen into stuff that we do, just link us up. I think your small help will go a long way to help us sustain what we are doing.
That is true. Yeah, we would definitely appreciate any help that we're able to get, whether it's physical or not, or funding as well, or just your time. I think that definitely all very much appreciated. Yeah, so I'd like to wrap up and say thank you so much to everyone for making time to share, to listen in on this webinar. We hope you've learned a few things. If you have any further questions, please feel free to reach out to us. We will definitely help connect you with Dr. Wan or Fathan if your questions are for them, or as well as Boon if you have any questions about citizen science. For those who might be, if you're listening in and you're international as well, and you're curious about how to kickstart a citizen science program, yeah, we'd love to get in touch to also maybe just give you a little bit of guidance there. Yeah, but thank you so much for everyone again for making time. We truly appreciate it, and we hope to see you out on the Bukit Kiara trails. If as a citizen scientist, lots very, very cool when you see in person actually. Thank you and have a very good night.